So the back half of this lecture is going to focus on glycogen metabolism and how that relates to glycogen synthesis as well as glycogen breakdown within skeletal muscle. So when we're talking about the regulation of glycogen metabolism and we're thinking about glycogen synthesis, we're thinking about insulin. So insulin is an anabolic hormone, meaning that it's promoting the storage of glucose in the form of glycogen. The enzyme that facilitates this is glycogen synthase. And when we're thinking about glycogen breakdown, we're thinking about skeletal muscle contractions. So we need to make energy to support mechanical work and transport. So basically muscle contractions, we're breaking down energy. We need to replenish that energy by breaking glycogen down into glucose, making glucose into ATP. So when we think about this, we're thinking about the enzyme glycogen phosphorylase. Importantly, both of these enzymes, glycogen synthase and glycogen phosphorylase, undergo covalent modifications. If we think back to last week's lecture, these covalent modifications are manifested by phosphorylation reactions, either turning on or turning off that enzyme, and this is our coarse level of tuning. So thinking about glycogen synthesis, the rule of thumb is that glycogen synthase, which promotes glycogen synthesis, is phosphorylated, less active. Glycogen synthase can be phosphorylated by a number of different enzymes, but the most known regulator is glycogen synthase kinase 3. So what actually happens is this glycogen synthase kinase 3, or GSK3, can phosphorylate glycogen synthase into its inactive form. If glycogen synthase is inactive, we're impairing or slowing down glycogen synthesis. If we're thinking about glycogen synthesis in the regards to insulin, what we actually see is that glycogen synthase kinase 3 itself can be regulated in the sense that GSK3 is also phosphorylated less active. So we take this bigger picture, we think about insulin being this anabolic hormone. So insulin wants to promote glycogen synthesis. Insulin, we go through that insulin signaling cascade. Eventually we produce AKT, or protein kinase B, whatever you want to call it. AKT is then able to actually phosphorylate or promote the phosphorylation of GSK3, making it inactive. If GSK3 is inactive, it's unable to turn glycogen synthase into that inactive form, leaving it in the active state. If glycogen synthase is in the active state, glycogen synthesis is enhanced or increased capacity for glycogen synthesis. So what we're thinking of here is inhibiting the inhibitor. On the other side of the equation, we can have insulin playing a role on promoting glycogen synthesis through the activation of glycogen synthase through its dephosphorylation. This dephosphorylation is promoted by protein phosphatase 1, which increases glycogen synthesis through activation of glycogen synthase. Importantly, the role of insulin in stimulating glycogen synthesis is highest immediately post-exercise. So we're thinking about replenishing those glycogen stores that we've tapped into during an exercise bout. So when we think about glycogen breakdown, again, we're thinking about skeletal muscle contraction, liberating glucose so that we can use it to make energy to fuel those skeletal muscle contractions. Importantly, glycogen phosphorylase is the major enzyme responsible for glycogen breakdown. And the rule of thumb is that glycogen phosphorylase is phosphorylated more active, meaning that this enzyme GPK, so glycogen phosphorylase kinase, is able to phosphorylate glycogen phosphorylase, making it more active. Moreover, muscle contraction, so increases in calcium levels, as well as epinephrine, can trigger the phosphorylation of glycogen phosphorylase kinase, meaning that it's even more active promoting glycogen breakdown in order to make glucose and making that glucose to break down further into ATP. So how does this actually happen? When we take a closer look at glycogen phosphorylase kinase, what we see is that it's comprised of 4 subunits: an alpha, a beta, a gamma, and a delta subunit. So when we're thinking about skeletal muscle contraction, we have to think about the fact that we have increased calcium levels. Importantly, the delta subunit of calmodulin is calmodulin on this glycogen phosphorylase kinase. So during skeletal muscle contraction, calcium can bind to that delta subunit, which is calmodulin. Binding of calcium to calmodulin activates glycogen phosphorylase kinase. We know the activation of glycogen phosphorylase kinase promotes the activity of glycogen phosphorylase to cause glycogen breakdown. On the other side of the equation, so skeletal muscle contraction and increased calcium was one side. The other side is epinephrine. So this is kind of our stress response that we'd see during exercise as well. So activation of beta-adrenergic system, basically, so epinephrine binds to beta receptors. And what happens is that binding to beta receptors causes an increase in cyclic AMP. Cyclic AMP is a second messenger, can go through its signaling cascades to activate PKA, and PKA can then phosphorylate glycogen phosphorylase kinase. This phosphorylation of glycogen phosphorylase kinase increases its activity, which then also increases the active form of glycogen phosphorylase to increase glycogen breakdown. So if we put this all together and we take a closer look, what we see, we've got these two separate pathways. The first being the contraction-mediated pathway, where skeletal muscle contraction, we have increased calcium levels, increased activation of glycogen phosphorylase kinase. On the other side of the equation, where we have epinephrine binding to beta-adrenergic receptors, eventually causing the phosphorylation or activation of PKA. PKA can then activate glycogen phosphorylase kinase. So now that we know both calcium and epinephrine can activate GPK, it's important to consider that during an exercise scenario, we would have both the activation of epinephrine and calcium. So with both epinephrine and calcium levels increased during skeletal muscle contraction, we can see that there's this additive or synergistic effect on the activation of glycogen phosphorylase kinase. So it's most active with epinephrine and calcium modulators kind of at play. So now we need to think about the other side of the equation, so stopping glycogen breakdown. So when glycogen breakdown is no longer required, so we're thinking at the end of exercise, we have this decrease in epinephrine levels. We also have this decrease in calcium levels, signaling for us to decrease glycogen breakdown because we're no longer exercising and we no longer require these elevated levels of ATP. So remember, glycogen phosphorylase is the major enzyme responsible for glycogen breakdown. So in order to turn glycogen phosphorylase into its less active state or its inactive state, we need to dephosphorylate it. And the way that we dephosphorylate glycogen phosphorylase is through protein phosphatase 1. When protein phosphatase 1 dephosphorylates glycogen phosphorylase, it's in its inactive form, and glycogen breakdown is decreased. So now that we've talked about the ways that we can regulate glycogen synthesis as well as glycogen breakdown, it's important to think about, once we've broken down that glycogen to liberate glucose, how does glycolysis fit into this picture, and how do we use that to then generate ATP to fuel our muscles' work? So this slide is majorly important because it's showing us the major ways that we can regulate glycolysis, or the entry into glycolysis. So we can get G6P from two sources, whether that's glucose uptake from skeletal muscle contraction or insulin-mediated pathways, or the breakdown of glycogen to liberate glucose. Essentially, I have the star beside glucose 6-phosphate because this is the committed step of keeping glucose within a skeletal muscle. Once glucose is turned into glucose 6-phosphate, it cannot leave skeletal muscle, unlike the liver where we talked about G6-phosphatase being able to convert glucose-6-phosphate back into glucose. So again, once glucose is turned to glucose-6-phosphate in skeletal muscle, it cannot leave the skeletal muscle. It is trapped here. So if we take the glucose uptake side of the equation, what we can see is that a hexokinase reaction results in phosphorylation that turns glucose into glucose-6-phosphate. This requires 2 ATPs because it's another level of control that allows us to commit glucose into its degradation into eventually pyruvate. The next level of control I want to talk about is phosphofructokinase. So this enzyme allows us to commit glucose to degradation. So the conversion of fructose-6-phosphate into fructose-1,6-bisphosphate is facilitated by phosphofructokinase. Importantly, phosphofructokinase is allosterically inhibited by ATP, meaning that if we have too much ATP built up, it's a signal for our body that we don't need more glycolysis to occur, so we can inhibit at this point in time in the glycolysis pathway. And another thing to remember is that all of this is occurring within that cytosol. So taking a step back and thinking about allosteric regulation, we talked about this last week. So when a ligand is bound to the enzyme, it allosterically regulates that enzyme, in this case of ATP. It's reducing its affinity for the substrate, which is fructose-6-phosphate. So again, in this example, ATP is our ligand. It binds to our enzyme, which is phosphofructokinase. What it does is it causes a conformational change, changes the shape of the binding site for fructose-6-phosphate, making it harder for fructose-6-phosphate to actually bind, decreasing the rate of this reaction. So what we see here is that when ATP levels are low, so we need glycolysis to break down glucose to make pyruvate and later ATP, we have basically increased PFK activity to promote this pathway to occur. But when ATP levels are high, we see this rightward shift where we require a higher concentration of the substrate, fructose-6-phosphate, in order to get the same amount of the phosphofructokinase reaction to occur. So basically, we've caused this conformational change. It's inhibiting the binding of fructose-6-phosphate, making it harder for fructose-6-phosphate to bind to the enzyme, meaning that we have a slowing of this reaction when ATP levels are high. So the last thing I want to highlight here is kind of the net result of glycolysis in that we yield 2 pyruvate molecules. So you'll remember that eventually we split into 2 glycerol 3-phosphate molecules, which allows us to make 2 pyruvate as our net outcome of glycolysis. Alongside this, we have a net gain of 2 ATP as well as 2 NADH, which are reducing equivalents. So as I just mentioned, the end of pyruvate, or the end of glycolysis, is the production of 2 pyruvate. Basically, pyruvate has 2 fates then. It can either be converted into lactate via lactate dehydrogenase, or LDH is its name. This is its anaerobic fate. Otherwise, it can be converted into ACETYL-COA by pyruvate dehydrogenase. This is its aerobic fate. So we'll start off by talking about lactate dehydrogenase, or the anaerobic side of the picture. So first off, it's estimated that 75% of lactate produced by skeletal muscle is actually converted back to pyruvate by neighboring oxidative muscle fibers. This is occurring through a lactate shuttling mechanism that we'll discuss on the next slide. So this relies on 2 key kind of rules. The first one being that LDH, its reaction is reversible, meaning that pyruvate can be turned to lactate through LDH, and lactate can be turned to pyruvate through LDH as well. This is a near equilibrium reaction that is governed by, again, its concentration of substrates and products. So more pyruvate will push the equation towards production of lactate, whereas more lactate will push the equation towards the production of pyruvate. The second one being there's differential expression of lactate transporters depending on the muscle fiber type. So MCT4 is on glycolytic fibers and it's related to lactate export, and MCT1 is on oxidative fibers related to lactate import. So here we see in this light colored box at the bottom, fast glycolytic fibers have less mitochondria and therefore they have a decreased capacity for aerobic production of ATP, meaning they have a decreased ability for pyruvate to be pushed to acetyl-CoA to go through the TCA cycle and through the electron transport chain. Instead, what happens is pyruvate through lactate dehydrogenase is converted into lactate in these glycolytic fibers, and we then require a shuttling mechanism to convert this back into pyruvate at the oxidative fiber. So what we see is that, again, the pyruvate is being pushed to form lactate through lactate dehydrogenase in this glycolytic fiber. And essentially, it's shuttled out of that glycolytic fiber through something called the MCT4 transporter, a lactate transporter. It's picked up by neighboring oxidative fibers through the import of lactate through the MCT1 shuttle. So again, noticing the differences in these MCT transporters between glycolytic and oxidative fibers, and noting the differences in their functions as well. Once lactate is brought into that oxidative fiber, what can happen is that now we have a buildup of lactate, which is going to push the LDH reaction towards pyruvate. Because oxidative fibers have a greater number of mitochondria, we're able to then take that pyruvate, convert it to acetyl-CoA, and push acetyl-CoA through the TCA cycle and the electron transport chain to make ATP. So now considering the aerobic side of the picture, so turning pyruvate into acetyl-CoA through pyruvate dehydrogenase, and noticing that this occurs within the mitochondria. So pyruvate dehydrogenase is a gateway for aerobic oxidation of glucose, so meaning that once we have glucose broken down into pyruvate, we can send pyruvate through oxidative metabolism, through that TCA cycle, and through the electron transport chain by converting pyruvate to acetyl-CoA. Pyruvate dehydrogenase, again, what it does is it catalyzes that one-way irreversible formation of acetyl-CoA, essentially through pyruvate into acetyl-CoA. And this occurs via decarboxylation of pyruvate and the generation of NADH. Pyruvate dehydrogenase can be modified by both covalent and allosteric modifications. So PDH is a multi-enzyme complex, so it has 3 enzymes and 5 coenzymes. And what PDH essentially does is it regulates the flux of carbohydrate oxidation, meaning that it's the bridge between carbohydrate and fat oxidation because it's allowing pyruvate to be turned into acetyl-CoA and entry into that TCA cycle. What I want to note here is from the Howlett paper is that PDH activation increases with exercise intensity. So as exercise intensity increases, we need to produce more energy through aerobic glycolysis, meaning that we need to break down glucose into pyruvate, and that pyruvate needs to be pushed through to acetyl-CoA in order to make ATP to fuel that exercise. When we consider the regulation of PDH, we have to think about the fact that PDH is again subject to those covalent modifications. So it has 3 phosphorylation sites, 1 inactivates the enzymes, and 2 act as barriers to hinder the activation of the phosphatase side of things. So the rule of thumb is that PDH is phosphorylated less active. So meaning that when it's phosphorylated, it's turned into its less active form, and this occurs through something called pyruvate dehydrogenase kinase. On the other side of the equation, PDH is dephosphorylated more active. This occurs through something called pyruvate dehydrogenase phosphatase. So there are 4 pyruvate dehydrogenase kinase isoforms, PDKs 1 through 4, and there's 2 pyruvate dehydrogenase phosphatase isoforms, PDP 1 and 2. PDK4 and PDP1 are the predominant isoforms found in skeletal muscle, and what's important to know is that these regulators, as well as pyruvate dehydrogenase, are found within the mitochondria, which makes sense because this is the committed step to bringing pyruvate into the TCA cycle by turning pyruvate into acetyl-CoA, committing it to the TCA cycle. Another point I want to touch on is during muscle contraction, we talk about this increase in calcium levels. Interestingly, a small amount of calcium actually can enter the mitochondria. And this small amount of calcium entry, so with skeletal muscle contraction, we have breakdown of ATP, the need for energy. It's basically thought to activate pyruvate dehydrogenase phosphatase and inhibit pyruvate dehydrogenase kinase, meaning we're increasing the activity of pyruvate dehydrogenase. So again, it's promoting this movement of pyruvate to acetyl-CoA to go through the Krebs cycle to make larger amounts of ATP. So now we need to consider the fate of acetyl-CoA. So just thinking about the TCA cycle and the electron transport chain, specifically how these reside within the mitochondria and how this is going to produce energy to fuel the work that we need to perform. So acetyl-CoA, we mentioned that it enters the TCA cycle. The goal of the TCA cycle is to produce reducing equivalents. So the reducing equivalents we're thinking about are NADH and FADH2. Another bonus of the TCA cycle is that it produces a small amount of ATP, but again, the major role of the TCA cycle is to produce reducing equivalents. So NADH and FADH2 are the reducing equivalents we're talking about that are produced through the TCA cycle. They are high energy electron carriers, meaning that when they enter the electron transport chain, they're actually oxidized. So NADH is oxidized to NAD+, and FADH2 is oxidized to FAD. The oxidation of these reducing equivalents releases free energy and ultimately results in the pumping of protons or hydrogen ions into the intermembrane space from complex 1, 3, and 4. What we see is that the oxidation of NADH results in the pumping of 10 protons from complex 1, 3, and 5 combined. And because FADH2 enters the electron transport chain at complex 2, we miss out on the protons pumped at complex 1, meaning that only 6 protons are pumped per FADH2, and this occurs at complex 3 and 4. So what we've done now is created this proton gradient in the intermembrane space, and that proton gradient is actually used to drive ATP synthesis through ATP synthase or complex 5 of the electron transport chain. So these hydrogen ions or protons travel down that ATP synthase and can actually convert ADP into ATP. So 4 protons are needed to create 1 ATP. So if 10 protons are pumped per NADH and it requires 4 protons to create 1 ATP, each NADH can generate 2.5 ATP. Similarly, thinking about FADH2, if only 6 protons are pumped per FADH2 and 4 protons are required to make an ATP, each FADH2 can yield 1.5 ATP. So the total net yield of ATP from glucose oxidation, or the oxidation of a single glucose molecule, is 32 ATP, which is pretty impressive. But what we'll see with fat metabolism is that the oxidation of a 1-palmitate molecule can generate a lot more ATP than this. So next Thursday, we'll talk about study design and research methods. This will be super helpful for interpreting and understanding the papers for presentations. And on Monday, we'll have a lecture similar to today's, talking about, on the other side of things, lipid metabolism.